happens at the promoter? The replication complex, right, the transcription complex, not the replication, but the transcription complex binds to that promoter region. So those sequences in here, and these consensuses are involved in binding up the promoter. The sigma factor recognizes these regions, sigma in particular recognizes this top box. So what is the function of consensus? Why aren't they identical? Why aren't they all the same? Well, remember that the sigma factor, or the factors that bind to these DNAs, actually, I guess you can't remember, I haven't really told you, but they recognize and bind most tightly to the consensus sequence. So a perfect top box is not bound very tightly. On the other hand, as one begins to stray more and more from that thing, like this CAT, GAT right there, same thing over here, as you get further away from the consensus, then the affinity of the proteins that recognize and bind to it go down. Right? So you have high affinity, low affinity promoters. At a high affinity promoter, there's a very high chance that that consensus sequence is going to be recognized by a transcription complex. The complex is going to bind and transcribe. At the lower side, the lower affinity side, the lower consensus sequences that are the promoter regions that are the furthest from the consensus sequence, sigma recognizes them much less frequently, binds less frequently, and transcribes less frequently. So the consensus sequence, for one thing, determines the rate of transcription, particularly in prokaryotic cells, but it's also true in eukaryotes. Okay, it's less true in eukaryotes because of all the transcription factors that we'll talk about that are also needed for the process. But here, these guys are that. There's another promoter region called the upstream element. It resides about 20 nucleotides outside of this region here, and it also is involved in that process, right? This upstream element is recognized by a region of polymerase in eukaryotic cells called the CTD. When we talk about eukaryotic polymerases, I'll tell you about that, but that UP region is upstream about 20 nucleotides or so. You come back and look at this slide to see it. Okay. So transcription elongation stage here, this is a process that shows you, I told you that you open a bubble, it's called the transcription bubble often, and that bubble is about, it opens, it can be around 25 or so nucleotides in E. coli or longer. Here's the hybrid with the duplex between the newly synthesized RNA, the new guy, and the template, this guy there. There's the duplex. That duplex is about 12 nucleotides or so. I told you that you need a helicase to open this, and you need gyrases, right? Topoisomerase. You can see what's happening here is you're moving positive supercoils in order to generate tension to open this up and allow it to align. And here it removes negative supercoils, which allows this in the back end to rewind again, but you need gyrates on both sides. Unwind it, rewind it. How about termination? So in elongation, I'm not going to say much about that. I told you because elongation continues as long as that duplex is formed. So termination, which is the third part, initiation, elongation, termination, the third step in transcription. By the way, remember in replication, it's the same thing. We talked about initiation of DNA synthesis where the promoter, where the replication complex all sets up. Elongation where the forks elongate, and then termination where synthesis stops. Same thing here. Termination. There's two types of termination that occur in bacterial cells. They're called row-dependent and row-independent. And row-independent is represented here. And row-independent transcription or transcriptional termination in bacterial cells is dependent on our sequence, which is the termination sequence that occurs at the end of the transcription cycle. If you look at this termination sequence, what you see is there is a repeat region, an inverted repeat region, right? CCT, CGA, CCT, hydrogen bonds so they don't form a very good complex. Everybody still listening? So what we need to do is we need to locate this within that 12 nucleotide or so duplex. And can't believe I have. So what happens is that remember we have these 12 nucleotides in here, and then we have this region in here. Okay, this region right here. So the 12 nucleotides will encompass, right, this region right here. And what happens is as that thing goes, it forms a so as that thing forms a hairpin loop, you have your RNA like this. Here's your template, right? And here's your RNA. Here's your 12 nucleotides. So when you get to that hairpin loop region, what happens is you have hybridization like this, right? But then you have these hairpin loops. All right, so these things here, which would be part of this hybridization here, form intramolecular bonds rather than intermolecular with the template. Y'all see that? Does that make sense? This is where you would have a bubble, right? This is your replication bubble in the DNA. So where there should be, all of these should be in duplex to hold this RNA complex. What happens is because of that, inverted repeating forms this intramolecular loop, which only leaves a few, simply less than 12 nucleotides to form this duplex between the template and the RNA. Everybody see that? So now there's very few. Not only that, but what are these guys here to the left? What are those? So, if you look at the picture right, this region here spans right here. So those guys are in duplex like this, which leaves these 5 or 6 or whatever in duplex. So these guys, this region here forms an intramolecular bond, which pulls some of these out of that duplex. They're up in there, right? Normally this region here would also be in duplex, forming a total of 12. Form this loop like this, that means only these guys in duplex. Not only are there only 6 or 7 of them, but they're also AT residues, so they have only double bonds. And essentially the RNA just rips away from the template. When it does, termination stops. Does anybody have a question? Rho-dependent is similar, but it involves a little protein that's called the rho protein. And the rho protein, or the rho factor, is a hexamer. And what it does is as RNA synthesis in a rho-dependent termination, as RNA synthesis goes on, you can see those guys are in hybrids in this RNA complex. What happens is that the rho factor actually jumps onto the end of the nascent RNA chain. So rho is an ATP-dependent helicase. You can see, look, it's a hexamer again. Requires ATP, unwinds this thing, but what it does is it binds to GC-rich regions at the end of an RNA, and then it runs up. It's able to move up that RNA molecule, and it moves faster than transcription. You can see it actually is moving up the tail there, right? And what it does is when it gets into the region that's in duplex, the helicase actually unwinds it. So rather than having this duplex come apart because of the inherent sequence in the RNA of the nascent RNA, this helicase actually runs all the way up the chain and starts to unwind this itself. And as it unwinds it, then the RNA